Okay, so our group went to Totland. Totland is located south of Bergen and it's a scarcely populated area, about 45 houses, including a school. And as you can see, it's also an agriculture area. We went there to have a look at the wastewater treatment plant. And as you can see here, it's just located nearby the river. We wanted to check the wastewater treatment plant efficiency and the impact on the Totland River where the effluent is connected. So the wastewater treatment plant, very simplified, 3 steps. The first one is physical. Here suspended particles are being filtered away, and the second one is biological. Here microorganisms deplete up to 90% of the organic matter, and in order to do so the water is being aerified. And the last one is chemical. Here aluminium sulfate is added in order to deplete the phosphorus, and along the way you have the sedimentation as well. Inside the plant, don't worry, it didn't smell as bad as you would think. Behind the blue box is roughly the outlet, and where we're standing to the left is the inlet. That morning there was a pipe burst, and our representative from Bergen Kommune was really worried about our results due to the pipe burst. That leads us to our result and to our materials.
So I'll tell you a bit about TCB, or thermotolerant coliform bacteria, which here is illustrated by E. coli, which is the most common form of TCBs, and they indicate fecal contamination of water. So we went to Totland and we took samples upstream and downstream of the outlet, as well as at the outlet itself. We took 4 samples of TCB at each site, and we had one large problem here as well, and that was that the outlet was 10 meters upstream from where we originally thought that it was. However, we sorted this out and we continued to take samples at the plant itself, at the outlet and at the inlets. After the samples were analyzed, we compared them with previous years to see if there was any difference between the years.
Sure. So phosphorus is another thing that we measured, and this is an important indicator of water quality because a high level of phosphorus in the river can lead to increased bacterial growth and also increased algal growth, and in extreme cases this can lead to eutrophication. So we measured this in a very similar way to TCB, and we took water samples upstream and downstream of the outlet, and also within the wastewater treatment plant. Different to the TCP, we only took 1 sample at each site because phosphorus is more stable than TCB. So this involved putting on waders and getting into the river, getting pretty cold, pretty wet. And then in the wastewater treatment plant, we only took a sample in the inlet and not in the outlet.
Yes, the bent examples. Pollution has been shown to affect biodiversity, and we took bent examples to assess the effect the plant has on the invertebrate biodiversity in the river. It was taken on all the same locations as the phosphorus were, using the kick sampling method, where we used this bucket we can see up here, placing it on the substrate and kicking sediment into it. These contents were then taken back to the lab to be analyzed. Biodiversity is affected by water quality, and here we see the 3 groups present. There was the Insecta, Oligochaeta and Platyhelminthes, and these symbols represent water quality. We will use 3 different methods to measure the biodiversity, and that was the BMVP and the ASTP, which shows the pollution tolerance, and the Shannon-Wiener index that shows the evenness among the taxas. And as we can see here, Oligochaeta and Platyhelminthes has high pollution tolerance, which would mean that if there was a lot of those, some places in the river compared to others, this could indicate a decrease in water quality.
So that leads us to our results and discussion.
Yeah. So I'll tell you a bit. I looked at the TCB values upstream and downstream to see if there was a difference. And this little tick mark in the corner here means that there is a statistical significance, while if there's a cross there, there's no statistical significance. So the downstream values we found to be significantly higher than the upstream values, And why, do you ask yourself? Well, we looked at the plant itself, because this would lead to excessive nutrients downstream, which again could affect the TCB values. This could be further enhanced by the pipe burst on the day itself. And this is also illustrated by the really high levels of TCB within the plant. They were so high that we didn't even get them. However, the values in the river were still low, so it wasn't really a big problem. We also looked to see if there was a significant difference between the years, and there was. And then we looked at why is there a difference between the years. We looked at rainfall. We had some statistical issues here because we hadn't learned how to use the right method when we were supposed to do it, so we needed to wait a couple of days before we could actually manage to do it. But what we did was that we looked at the average rainfall from the 2 days before the sampling, as well as on the day of the sampling, and we measured this against the TCB values of the different years. And there was not a significant interaction here, but as you can see, it's a clear trend towards increasing TCB values with increasing rainfall. And our year, the 2012, was not really going together with the rest of the results, but I'll tell you more about this later. So, why does the rainfall affect the TCB values? Well, there is grazing and agriculture here, so this could lead to more nutrients in the river when it runs down when it rains. And therefore the TCB values could also be increased in the river. And why was our year so much different from the previous years? Our only explanation for this is that there could be less grazing.
So the results for the phosphorus are quite similar to TCB in the fact that they show a higher concentration of phosphorus downstream compared to upstream. However, this was not a significant difference, and that's surprising when you look at the graph because it seems like it's significant. And this insignificance is likely due to the small range in phosphorus concentrations. You can see it ranges from just 14 to 21. And also, we had a small sample size, so we were just comparing 3 sites downstream to 3 sites upstream. This isn't really enough for the significant test. You can also see that the level in the wastewater treatment plant was a lot higher than the level in the river. And this shows that between the inlet and by the time it reaches the river, the phosphorus level has been reduced a lot. So the next thing we looked at was whether there was a relationship between TCB and phosphorus. And as you can see in this graph here, there is a relationship which is significant, and as phosphorus increases, so does TCB. And this is what you'd expect to find because phosphorus increases the growth of bacteria. So on the day that we sampled, as you've heard, a pipe burst within the plant. And this was really bad for us because it meant we couldn't measure the plant's efficiency because it wasn't fully functioning. However, we did get data from Bergen Kommune from May to July of this year, so our study was in September, so just a couple of months earlier. And from that, we were able to calculate the phosphorus removal within the wastewater treatment plant. And as you can see here, in all 3 of the months, the removal was greater than 90%. And 90% is what the Norwegian government sets out as the requirement for wastewater treatment plants. Therefore, it shows that the wastewater treatment plant under normal conditions is functioning correctly. So these results, coupled with the results we found with low concentrations within the river, showed that phosphorus isn't a cause for concern at Totland.
Yes, the Bentic results. Here we see the same table as last time, and you can see here that we also have the number of individuals, and we see that those insecta, those of intermediate pollution tolerance, dominate. We also have these pie charts that shows us the percentage of pollution tolerance among all the taxa, and we see that downstream 2/3 are of intermediate tolerance, while upstream over 3/4 are of either intermediate or low pollution tolerance. The results show both good and bad water quality depending on the indexes used. But both pollution tolerance indexes and the evenness index all show a decrease from upstream to downstream. But again, the results are considerably biased due to a lot of problems with the sampling method and a lot of difficulties in the lab. We lacked both expertise, time, and experience. There was a lot of frustration in the lab. So overall, the results are too skewed, too biased, and too small.
So, our conclusion and the bigger picture.
From our study, comparing between the upstream and downstream of the river, the phosphate level was not significant. Our TCV was significant, but the TCV value was low. Thus, it reflects moderate to high water quality. And the benthos result shows some trend. However, due to sampling bias, our results were skewed. Despite the pipe burst the very day, we've still got some good news for the Totlanders and the people downstream, that the water quality of the effluent was not compromised and the wastewater treatment plant works efficiently. During the implication of our study, this type of plant, which is simple in design, works efficiently and is cost effective, can be replicated to other areas, such as in countries like Nepal, where wastewater treatment plant is almost non-existent, where above 40,000 children below 5 years die of waterborne disease such as diarrhea and cholera, where major water systems are used as sewage disposal sites. This type of plant can be promising to these areas to resolve their water sanitation problem.
We would like to thank Karen and Becky, and also our representative from BIO300, Rune Fredrik. Thank you for listening.